Okay, so we are going to start with topic 1.1, statistics, and we are going to see what statistics is about. Statistics, first of all, is the science of data. And if you saw the introductory video, you saw that data can give us a lot of information about a lot of things, but statistics gives us a way to analyze this data to make it mean something. Okay, so we are going to start by looking at what is data. Data is information, and it can be in the form of facts or figures obtained from experiments or surveys, and this data is used as a basis for making calculations or for drawing conclusions. This, by the way, is the definition of data obtained from the Encarta dictionary. Okay? Now, why do we need to use statistics in science? Data can be collected, for example, about a population. If you run a survey, how many pets do students have in general in school? Or what is the favorite color of girls in ages from 6 to 12 years old? Those kind of information. Okay, data can also be collected about a process, for example, in an experiment. If you have a plant and you want to see how much temperature affects the rate of photosynthesis, you can do an experiment and then you can collect data. You can increase the temperature to see how much this temperature affects the rate of photosynthesis, and then you can collect data about how much photosynthesis has occurred at the different concentrations. So based on this, we can say that there are two types of data. There is qualitative data and there is quantitative data. First of all, qualitative data is information that relates to a characteristic or a description. We could say observable qualities. When we are talking about characteristics, this information is often grouped by descriptive category. That means, as an example, what type of plant is it? What type of insect? What color is it? If you're comparing the leaves of a plant that is in the shadow versus the leaves of a plant that is in the sunshine, you can see that the color of the leaves are going to be different. They are obviously going to be green, but they are going to be a different shade of green. So what shade of green is it? That is also considered qualitative data. And for example, the rank of flavor in a taste testing. Okay, for example, is it sweet? Does it taste like apples? Is it a taste that you can recognize? Is it salty? Is it more salty than before? Is it less salty? And so on. Okay, so all this qualitative data, it talks about descriptions. Okay, qualities that can be described as something, but that they are not necessarily evaluated numerically. Okay, you can have for example, different shades of green and give each shade a numerical value. And so you could record your shades of green with a number, but originally the type of data is qualitative. Okay, you're talking about the color. You're not necessarily talking about a number. On the other hand, before we move on to quantitative data, qualitative data can be manipulated numerically. Even though we are talking about descriptions that do not have a number, we can still manipulate it numerically. We can take a percentage, for example. In this case, we have the results of a survey about what teens think if there is a need for an environmental action or not. Okay, here we have the results. Some of them are neutral towards the need of environmental action. Some of them agree that there is a need for environmental action, and some of them strongly agree. So even though your answers are not numerical, they are qualitative, you can still tally up the numbers, you can still take percentages, you can add them, you can subtract them, and so on, and they can be manipulated numerically. Okay? Quantitative data, on the other hand, is a measure that is used or that it occurs naturally using a numerical scale. That means that these things that you are measuring, they automatically come in the form of a number. For example, if you're measuring how long is something and you're using a ruler, you can see that it measures 9, maybe 10, and if you grab the next one, it doesn't measure 9, it measures 8, and so on. So these come naturally in the form of a number. Examples of this are, for example, chemical concentration. How many moles per liter are there in your concentration of salt? What is the temperature of the room? What is the length of the size of a tree? How much do you weigh? And so on. So these types of data that you can obtain that comes naturally in the form of a number is considered quantitative data. Okay, now when we talk about quantitation, we are talking about measurement. And when you are collecting data for biology, this data must be measured carefully using laboratory equipment. For example, if you want to see what is the quality of the water in a pond in a specific ecosystem, you can use a pH meter, you can use timers, you can use thermometers, meter sticks, balances, pipettes, and so on. So all these instruments, all this laboratory equipment helps us to measure things, to collect data. Okay? Now, the limits of the equipment that is used, for example, if you use a balance, if you use a ruler, it has a limit, and this limit adds some uncertainty to the data collected. All equipment has a certain magnitude of uncertainty. That means that if you have a ruler that is mass produced, that means a regular ruler that you can purchase from any store, is this ruler good to measure things that are 1 centimeter big, for example? Most likely, yes, because in this ruler you have the divisions for centimeters and you also have the divisions for millimeters. So you can pretty certainly see if something measures 5 centimeters or 3 centimeters or 1 centimeter. Is it good to measure things that are 1 millimeter in size? Probably still yes, because you can still see the divisions of millimeters, so you can see whether it's 1 millimeter or 3 millimeters or 5 millimeters, so you can still use that ruler to a certain amount of certainty. Is it good to measure something that is 0.1 millimeter big? Probably not. The mass-produced ruler only has the millimeter markings. So if something is 0.1 millimeter big, it would probably be very difficult for you to measure that with a certain amount of exactitude with a ruler, a certain amount of precision. Okay, so this level of uncertainty, this magnitude of uncertainty, comes with the equipment that you use to measure. Okay, for quantitative testing, if you have to know really exact numbers, you have to indicate the level of uncertainty of the tool that you are using for measurement. So every time that you are in the lab, you are going to write down your data values. For example, my result was 5, okay, but 5 with the level of uncertainty. How close is it to 5? Is it 5.0, 5.1, 5.15, 5.154, and so on? Okay, so how exactly close to 5 is your data you will be able to tell by the instrument, but by the tool that you are using to measure. Okay, so again, what is this uncertainty business? Uncertainty means that you're not certain about something, but how is this translated into science and how can you calculate it? Okay, we talked about that all measurements are approximations. Okay, no measuring device, no matter how good it is, can give you perfect measurements. Okay, it can give you very close approximations. So if instead of using a ruler to measure something that is 0.1 millimeters big, you are using a micrometer that can measure up to 1,000th part of a millimeter, then you could probably measure 0.1 millimeters with no trouble. Okay, but remember that all measurements are approximations. Some are very close approximations and some are large approximations. Okay, but no measuring device can give you perfect measurements. They can give you very close approximations. And this not perfect approximation is called uncertainty. In this example, by convention, that means everybody agrees, okay? A mass is measured to be 13.2 grams. So something weighs 13.2 grams. And this 13.2 grams is said to have an absolute uncertainty of plus or minus 0.1 grams and is said to have been measured to the nearest 0.1 gram. In other words, we are somewhat uncertain about the last digit. It could be a 2, but then again, it could be a 1 or it could be a 3. Okay? This uncertainty is given to us by the number. Okay, we have 13.2 grams. So we know for sure it's close to 13. But is it close to 0.2? Well, it depends on what we measure it with. By the looks of this, if we just see the number 13.2, we can assume that it has been measured by a balance that only gives you one decimal number. So the level of uncertainty is plus minus 0.1 gram. It could be a little bit more than 0.2. It could be a little bit less than 0.2, but the balance is not going to tell us exactly because the balance can only give us one decimal place. So the level of uncertainty is plus minus 0.1 grams. Okay? Remember that this is a video, so if you didn't get this last part, you can always pause, you can rewind, you can watch it as many times as you want until it makes sense. Okay? So we take this other example. How can we determine uncertainty? Well, usually the manufacturer, the company that has made the instrument that measures specifically for laboratory equipment, is going to indicate the level of uncertainty. Okay, so you can always read what is provided by the manufacturer. In this example, you can see the picture. We have a graduated cylinder that has a capacity of maximum 250 milliliters and a minimum of 5 millimeters. Okay, right underneath that, and I wrote it in black on the side so that you can see better, it says in 20 degrees Celsius plus minus 2 milliliters. Okay, that means that if you put water inside of this graduated cylinder and this water is around 20 degrees Celsius, is not too hot, is not too cold, then the level of uncertainty is going to be plus minus 2 milliliters. That means that if you measure 200 milliliters with this graduated cylinder, you will have an uncertainty of 2 milliliters. It can be that means that it could be 2 milliliters more than 200, so 202, or it can be up to 2 less. That means 198. But we know with certainty it is around 200 milliliters. How much certainty? How certain are we? Plus minus 2. Okay, that means that our exact value is not going to be 200.0000000. No, why not? Because our instrument, our graduated cylinder, has an uncertainty of plus minus 2. So even if we measure very carefully to the line that says 200, there can be a slight variation. It can be probably 200.5 or 200.1 or 199.9. Okay, so we are going to know for sure that it is less than 202 and more than 198. That means around 200 plus minus 2 milliliters. Okay, so when you are determining uncertainty, be sure that the number of significant digits in the data table that you are writing down or in the graph that you are drawing reflects the precision of the instrument used. For example, if you are using a balance that gives you plus minus 0.1 grams and you took the average of your data points and you plot the average in your graph, you cannot have a number like 5.321. Why not? Because the precision of the instrument only gives you one decimal place. So there's no way that you can know for sure that it's 5.321 if the precision is only one decimal place. So it would only be 5.3. Okay, so make sure that always you're using the significant numbers and the precision and the uncertainty of your instrument that is consistent all the time. Okay, for example, if the manufacturer, here we can see in the example, the manufacturer states that the accuracy of a balance is to 0.1 gram and your average mass, that means your the mean and the average of the numbers that you have obtained using that balance is 2.06. You calculate all those and you take the average and your result is 2.06. Because your balance accuracy is to 0.1 grams, then you need to round the average to 2.1 grams instead of 2.06, okay? Your data must be consistent with your measuring tool regarding significant figures. Now, significant figures is a concept that you should know from math and you should know from physics, but if you don't, make sure that you review and make sure that you understand all the rules for using significant figures. The same rules that you use for biology are going to apply for math, for physics, and for chemistry. So if you don't know these rules, or if you don't remember significant figures, or if you never learned significant figures, you can use this tutorial in this web page, this refresher, to this right here, to understand significant figures and to know all the rules for rounding up or rounding down the numbers so that you don't get your answers incorrectly, or that your graphs make sense, or that your data is correctly processed. Okay? So significant figures is something very important, and as such we are going to be doing a few exercises in class. So please make sure that before we do those exercises, you know what significant figures are and what we are talking about. All right, so how can we find the limit of uncertainty in a measuring device? First of all, like we said, we can look at what the manufacturer is telling us. The manufacturer is going to tell us 200 plus minus 2 milliliters in the example of the graduated cylinder. But if we are using a ruler, just a regular ruler that you purchase at the store, is not going to come necessarily with the level of uncertainty. So how can you determine the limit of uncertainty if the manufacturer doesn't tell you? Well, one way is to use the smallest measurement unit. That means, what is the smallest number that you can see in your measuring device? What are the increments in your measuring device? For example, in a metric ruler, just a regular ruler, is lined to 1 millimeter. That means that all the little lines tell you up to the size that is 1 millimeter. So in this case of the metric ruler, the limit of uncertainty of the ruler is plus minus 1 millimeter. Okay, that is the smallest measuring unit. If the room temperature is read as 25 degrees Celsius and you use a thermometer that is scored at 1 degree intervals, so you can see the lines and each line represents 1 degree, then what is the range of possible temperatures for the room? You have 25 degrees Celsius and then plus minus 1 degree. So what is the possible range? It could be 24 or it could be 26. Plus 1 degree, 26, minus 1 degree, 24. Okay, so looking at data again. Let's assume that you have gathered data, you have done your experiment and you have your data. Now you need to know how accurate your data is. That means how close are the numbers that you got to the real results. And this accuracy is also known as bias. You can say how biased is your data or how accurate is your data. Okay? How precise is your data is also another important question that you need to ask when you are handling your data. There are a lot of test systems in statistics that have some uncertainty because this uncertainty is given by the measuring device, but you can estimate the limits of the experimental uncertainty, and that is very important for you to do. So, for example, if you go out to the jungle and you measure the height of the trees, that is very difficult to do. So, probably you're not going to have a very exact number, and you're going to have a lot of uncertainty. So you need to quantify how much uncertainty is there in your data. How reliable is your data? Can you really say that all the trees are the same height or that they are different heights just by looking at them? You probably cannot. You're not going to use a ruler to measure the how tall is a tree because they're just too big. It would take you forever. And so depending on the instrument that you use and depending how you measure how tall a tree is, you're going to be able to say that your data is more certain, more precise, or less precise. If instead of measuring trees, you are measuring, I don't know, the size of cells of trees, and you can use a microscope that has a ruler attached to it, a special ruler, a microscopic ruler attached to it, then you would probably be able to say with much more precision the size of a cell. Okay, now if we look at this diagram here, these four examples are going to give us the difference between accuracy and precision because they are not the same thing. Okay, accuracy and precision are not the same thing. Let's look at this picture square by square. If we start at the top left corner, we have the bullseye. Okay, that means the center is where you want to hit. Okay, but you took four measurements and you got all four different numbers. In this case, the top left corner, you have not accurate and also not precise. Why not? First of all the numbers are very different from each other. And also, all the numbers are different from the real number that is in the center. So in this case, you're not being accurate, and also you're not being precise. Okay? Now, if we move on to the right top corner, it says accurate, but not precise. What does it mean, accurate? Accurate means that all the numbers, even though they are different from each other, all the numbers are relatively close to the center. Okay? Well, maybe the one in the top and the one in the left are not so accurate, but the one at the bottom and the one on the right are a little closer to the center than before. So these numbers could be accurate, but they are not precise because each of them is different from each other. Okay? In the bottom left, we have not accurate, but precise. That means that the 4 times that you took the measurement, you got pretty much the same number, like, I don't know, 5.1, 5.2, 5.0, and 5.3, okay? However, it's not accurate because the real number was 2. So even though you got all 4 numbers that were very close to each other, that means that you measured in the same way all 4 times, you measured wrong in the same way all 4 times because the real number is 2 and you got 5.1, 5.0, 5.2, and 5.3. Okay? In the last bottom right corner, you have a measurement that is accurate and precise. It's accurate because all 4 numbers are in the center. They are close to the real number. And it is precise because there is not a lot of difference between all the numbers. So this would represent, if the real number is 2, this would represent 1.9, 2.1, 2.0, 1.8, or even 2.2, okay? But this means that they are both accurate and precise because they are close to each other. Okay, it's pretty much the same number all 4 times, and also it's close to the real value. Okay, all right, so why do we need to know this? We need to know how accurate our measurements are because we need to know really how close to reality we are. Okay, now usually when you're doing an experiment, you repeat it more than one time because maybe that one time that you measure is not close to the real result. So how can you know whether your data is accurate or precise if you don't repeat? So that's why in the lab we are always going to repeat things again and again and again. Okay, now in this example, okay, when we need an accurate measurement of something, we usually repeat the measurement several times and then calculate an average value because from all the times that we repeat it, we don't know which one is closest to reality. So we just take an average, we take the mean, we take the sum of all of them and divide by the number of times that we tried. Okay, so here in this example is a graphical representation of the data points that you got from an experiment. Okay, your measurements are the red dots over here, and the blue is the average value that you calculate from the red measurements. Okay, now We go through the same process for the next measurement. For example, we have the water from a pond and the water from a river, and we want to see if they are different from each other in, I don't know, pH value. Okay, so we measured the pH of the water in the pond before, and now we are measuring the water from the river, and we have these measurements, and we also take the average. Okay. Now we can compare the averages from the pond and the average from the river to see if they are very different. Once the two averages are calculated for a set of data, that means one average for the river and one average for the pond, the average values can be plotted together on a graph to visualize the relationship between the two samples, to see which one is bigger or smaller and so on. Okay, so this is our plot, this is our graph, and we have the average from the pond, average 1, and the average from the river, average 2. But how can we be sure that these values are really different? If you look at this graph, you can see that they are different. They're not the same value. But are they really that different? Maybe this represents a difference in pH of 5.4 and the other one is 5.2. Is that really different? Is that going to make a significant difference between them? Well, in order to know that, we can calculate and we can use statistics to find out. Okay? So perhaps in this case, our measurements are not very precise. So how can we know if they are really different? Okay? We can construct error bars to represent how much error there is in each measurement. Okay? One of the first things that you are going to see in the biology guide is 1.1.1, define error bars. Error bars are a graphical representation of the variability of data. That means with the error bar, you can represent graphically how much variation is there in your data, how different are your values from each other. Okay, and error bars can represent range. That means your values go from 5.0 to 5.5. They can represent standard deviation, which is something we're going to talk about in a little bit, or they can represent estimates of variability. Okay, we're not going to go into much deepness in that because that's more like mathematics and statistics per se. Okay, so here in this example, we have the samples from the pond and the samples from the river, and we can see with the error bars that, well, although they looked very different in the beginning, now that we have all the information with the error bars, they don't look that much different now, do they? Okay, so how do we draw the error bars? The simplest way to draw an error bar is to use the mean, that means the average, as the central point, and then use the distance of the measurement that is furthest from the average, or the mean, as the endpoint of the data bar. That means we have in the center the average value, and then we have all the numbers on the left-hand side, and then we have the value that is the farthest from the average. So that's the value that is further away from everybody else from the average, and we use that distance. Okay? The calculated distance between the value that is further away from the average and the average. And then in the bottom, we just repeat the same value. Okay? So in this case, if you look at this graph, what do error bars suggest? Okay? If the bars show extensive overlap, that means there's a lot of region that is the same between the error bars, it is likely that there is not a significant difference between those values. Okay, if you look in this graph, it says at the bottom in the x-axis you have Amazon, Sleeper, Rating Zone, Real, Movie Critic, and Friends. Okay, and the little squares represent the means, and then on the y-axis you have the values, the 95% CI percent total useful recs, whatever that means. Okay, but it's a value. So you can see the difference between Amazon and Sleeper. You can see that the means are not very different, and the error bars overlap extensively. So you could see that you could say that the difference between Amazon and Sleeper is not very big. There's not a lot of difference between those. What about the difference between Friends and Rating Zone? Those are probably two different ones because first of all, the means are different, but the error bars don't overlap at all. Okay? Let's look at more examples of this. If the error bars for two average values don't overlap, the conclusion that you can get from this is that these two values, those two averages, are different, and that is justifiable. Not only are the average points different, but also the error bars do not overlap. So none of the data points were the same. So you could specifically say that the water from the pond is definitely different from the water from the river. Okay, that is a justifiable conclusion. Before we move on, we are going to do a quick review of 3 ways to measure central tendency. What does central tendency means? It means where most of the data points are. Okay? One of them is the mode. The mode is a value that appears most frequently. If you're measuring how tall are, I don't know, the boys in the classroom, then there is going to be one value that appears the most. I don't know, maybe you have values that vary between 5 and 10. I'm totally making this up. Values between 5 and 10, and the number 7 comes up very often. So 7 would be the mode, would be the value that appears most frequently. Okay? Another way to measure central tendency is the median. That means when all the data are listed from least to greatest, for example, from smallest to biggest, the value at which half of the observations are greater and half are lesser. That means if your numbers are from 1 to 10, and you have 5 number 1s, 7 number 2s, 6 number 3s, 2 number 4s, and so on, you could probably find the number at which half of the observations are bigger than that number and half of the observations are lesser than that number. That doesn't mean necessarily that the number between 1 and 10 is going to be 5. Maybe there's, I don't know, 10 number 1s and then 1 number 2, 1 number 3, 1 number 4, 1 number 5, 1 number 6, and 1 number 10. So the number would probably, the median would probably be 3 or 4. Okay? Now if you have 2 of each, probably the median would be 5. Okay? And the most commonly used measure of central tendency is the mean, which is the arithmetic average. That means you sum all the data points and then you divide by the number of points. Okay, for example, if you took 3 quizzes and your grades for each quiz were 10, 20, and 15, you add 10 plus 20 plus 15 and then you divide that by 3, and that number that you get, that's the mean, that's the average. Okay? Now, using statistics for biology, let's look at a practical example. How can the leaf lengths, the length of a leaf of a tree, for example, be displayed graphically? How can you make a graph out of it? This is the example that we have. We take the leaves of a tree, and they are all different sizes, and we can measure them, and we can put it in a table, but how can we display this graphically? Simply measure the lengths of each leaf, and then we need to plot how many leaves are of each length. Okay, so here we have on the x-axis, we have the length of a leaf. So all the leaves that measured 1 centimeter are going to be in the column 1. All leaves that measured 2 centimeters are going to be in the column 2. So we can see that there's probably one leaf that measured 2 centimeters, and so on. Okay, so in this graph, we can see that the most common size of leaf over here was 10. That means that most of the leaves measured, 11, measured 11 centimeters long. Okay, and we can also see that there are only a few leaves, like 1, that measures 2 centimeters. Okay, here we can see that about 19 leaves measure 11 centimeters. Okay, this kind of graph is called a frequency diagram or a histogram. And a histogram is a classic bell-shaped curve. It has the shape of a bell, okay? And this histogram is also known as a Gaussian distribution curve. And it is also known as a normal distribution curve, okay? Basically what we are doing, we are representing all the sizes of the leaves of a tree in a way that can be easily seen which sizes are the most common, where is the mean, how spread out are the leaves? Are there very, very small and very, very big leaves, or are all of them pretty much the same size, and so on. Essentially, it means that in all studies with an adequate number of data points, that means all the experiments that you have more than 30 data points, okay, a significant number of results tend to be near the mean. That means if you're measuring a tree and you're measuring all the leaves, probably most of the leaves are going to be close to the mean. And there are going to be a few that are going to be smaller, and there are going to be a few that are going to be larger. Okay, but it can give you graphically an idea of what the distribution of sizes is. Now, standard deviation. The definition of standard deviation is that standard deviation is a statistical tool that tells you how tightly or how close together all the various examples are clustered around the mean in a set of data. In this graphical example at the bottom, we can see that the mean is the red point over here and the white is the data points. So if all the data points are very close to each other and they are clustered around the mean, they are very close together, that gives us one type of information. If the data points are not clustered around the mean, that gives us another bit of information. Okay? If they are clustered, we can say that the data points are more precise. And if they are not clustered, they are less precise. Okay? Now, standard deviation is a more sophisticated indicator than just the average. Okay, we can take the average, but the standard deviation is a little bit more sophisticated indicator of the precision of a set of given number of measurements. Okay, so you took the sizes of the leaves of a tree and you added them up, you did the histogram and so on, and now you can take the standard deviation. How far apart are the data points from each other? How precise was your measurement? Okay, let's take another example. You measure the temperature of the room and you measure it, I don't know, 10 different times just to make sure that it is what you think it is. Okay, now all the 10 times that you measured the room temperature, you got a slightly different result. Not very different, just a little bit different. Okay, and with those 10 data points and using standard deviation, you can have for sure the level of uncertainty that would tell you a number that can tell you really how different are the data points from each other. Okay, so the standard deviation is like an average deviation of measurement values from the mean. That means from those 10 data points, how far apart is each of those data points from the mean? Like we said before, if we look at the previous image here, clustered, if they are very close to each other, that means every time that you measured, you got pretty much the same result. Or not clustered, that means every time you measured, you got a very different number. Okay, that still gives you the same mean, but the variation is very large. Okay, let's go back to where we were. So a typical standard deviation curve looks like this. Okay, just like we saw before. And in this case, the mean or the average value is indicated by this black line in the center. Okay? Now the red area represents one standard deviation. What does it mean? It means that when you take the standard deviation of all your values, you're going to get one result. Let's assume that the result, the standard deviation that you calculated is 5. Okay? So one standard deviation would mean from the mean 5 units to the right and 5 units to the left. So the red represents one standard deviation. Okay? And if we take into account the green also, the green area, this would mean 2 standard deviations. That would mean 5 from the mean to the red and then another 5 from the red to the green. Okay? So why is this important? Because according to this curve, one standard deviation away from the mean in either direction, that means plus or minus 1 standard deviation, that means the red area on the preceding graph, on the graph before that we just saw, accounts for somewhere around 68% of the data in this group. That means that 68% of your data points are going to be between one standard deviation, plus or minus one standard deviation. Okay? Two standard deviations away from the mean, that means the red and the green, account for roughly 95% of the data. That means that 95% of the data is going to be plus minus two standard deviations away from the mean. It makes sense, okay? And finally, this is what the graph looks like. The red area is plus minus one standard deviation. The red plus the green is plus minus two standard deviations. And the blue is plus minus three standard deviations. 68% of your data is in the red area, 95% of your data is in the green area, and 99% of your data is in the blue, green, and red area. Okay? Three standard deviations account for about 99% of the data. So, how is standard deviation calculated? With this formula. Okay? This you have the square root of sigma. Sigma means the sum. X is every value. X with the bar on the top is the mean or the average of all the values. So you subtract each score minus the mean of the average, and then you square it, and then you divide by n minus 1. N is the number of values. That means if you measure 30 times, it will be 30 minus 1. Okay? So you do this with a calculator, and for each of the 30 values, and then you take the square root of that, and then that's the number that you obtained for standard deviation. Do you need to know this formula for the test? Well, not the formula, because the formula can be calculated with a scientific calculator, or it can be calculated with Microsoft Excel, and this is something that we are going to do exercises in class with your results from data. So you're going to go to the lab, you're going to obtain data, and then we're going to put that data on Excel, and then we're going to use the STDEV, the standard deviation formula, to calculate standard deviation. So you don't need to know the formula for the test. You can also try the different tutorials that you have online, like this one over here. But although you don't need to know the formula, you do need to know the concept. You need to know that standard deviation is a statistic or a statistical tool that tells you how tightly all the various data points are clustered around the mean in a set of data. Okay? When the data points are tightly close together and the bell-shaped curve is steep, the standard deviation is small. What does that mean? It means precise results. Okay? When the data points are spread far apart from each other and the bell curve is relatively flat, that means a large standard deviation, and that suggests less precise results. Okay? Here, let's look at this example. Which sample population has the longest mean bill and B, the greatest variability in data? This is a comparison of the bill length, that means the beak, el pico, from two hummingbirds, A. colubris and C. latirostris. Okay, so we have measured their beaks and we have taken the mean, okay, the average of the beaks, and then we plotted that in this graph. And we also included the error bars that represent the standard deviation of the population of A. colubris and the population of C. latirostris. Okay, so for question A, the longest mean bill, that means the biggest average. If we see the average for A. colubris is 15.9 and for C. latirostris is 18.8. So which one do you think has the longest mean bill? Which one has bigger bills? Okay? I'll give you 2 seconds to figure that out for yourselves. Write it down somewhere so that when I show you the answer, you can see if you were right. Okay? And for question B, which one has the greatest variability in data? Remember that the error bars represent the standard deviation in this case. C. latirostris has smaller error bars. A. colubris has bigger error bars. So which one of these represents more variability? Here are the answers. Okay, So the longest mean bill, C. latirostris, as you can see, 18.8 is definitely bigger than 15.9, yes. But which one has the greatest variability in data? We can see that the one with the bigger error bars, A. colubris, is the one that has more variability, okay? But are these results significant? Are they significantly different from each other? Is 18.8 really that much different than 15.9? How can we tell? We can look at the overlap of the error bars. If there is large overlap, that means lots of shared data. That means that the results are not likely to be significantly different. That means that the difference between means is more likely due to chance. Probably we chose the C. latirostris with the bigger beaks in general. From all the C. latirostris, we chose the ones with the bigger beaks. And it happens that we chose the ones with the smaller beaks for A. colubris. Okay, so if there is a large overlap, that means that they are not significantly different. That means that 18 is not too far away from 15. Okay? On the other hand, if we had smaller bars and the bars did not overlap at all, that means there is no or there is very little shared data. That means that the results are likely to be significantly different. That means that definitely 18 is different from 15, okay? The difference between means is most likely to be real. So based on this graph, you could say that C. latirostris in general have bigger beaks than A. colubris, okay? Another example, these questions. Also, I'll give you a second or two to write down which one you think the answer is before I show you the answers, okay? So in this case, we have set A and set B, and each of these have their bell-shaped curves, okay? So in this case, which one has a larger range? Which one has more variability, high variability, either A or B? Okay, based on the shape of the curve, you should know which one has more variability. Okay, you got the answer? All right, so question B, which one has a greater standard deviation? Which one has more standard deviation? Okay, you can assume that the mean is at the center of the curve. All right. Letter C, which one shows more precise results, either A or B? And letter D, which one has a higher mean, a higher average? Okay. And finally, question E, which one has a higher frequency at the mean? A or B? Okay, so here are the answers. A larger range, which one has more variability? Set B. Why? Because the size of the bell shape is bigger, is more flat, and is also more wide. Okay? Which one shows a greater standard deviation? Again, B. Why? Because the bell shaped is flatter. Okay? Which one shows more precise results? A. Why? Because the bell shaped is narrower, is steeper, and that shows us that there is less standard deviation, less variability, and therefore more precision, more precise results. Which one has a higher mean? You would say B, not because the curve is taller, but because you have to look in the x-axis, the continuous variable. Remember, the mean is usually at the center of the curve, and so the one towards the right has a higher value than the one towards the left. The one towards the left is closer to 0. So B, in this case, has a higher mean. Now, which one has a higher frequency at the mean? Set A. How do we know that? Because the y-axis is telling us the frequency. Okay? There are more values at the mean in A than there are in B. Okay? All right? So that is the end for today.